we looked at an image similar to this one previously where we kind of looked at what makes each blood type unique. In other words, what makes blood type A, and why is it different from type B or AB or O? So if you recall, the reason why each blood type is named what it is named, A, B, or O, is due to the presence or absence of the antigens on the surface of the red blood cells. So again, if we look at the type A red blood cell in the second column, we can kind of see that the blood cell has those circular antigens attached to the surface, but in B it has kind of the diamond-shaped antigens attached to the surface. Down in the bottom row, we can see that those red circles are identified as the A antigen, and the kind of turquoise diamonds are identified as the B antigen. So blood type A has A antigens, blood type B has B antigens. And then we see blood type AB, and that has both types of antigens. And then blood type O, no antigens, so it is blood type O. What we also need to notice is that blood type A has anti-B antibodies, or B antibodies, which means that if the B antibodies get mixed in with blood with the B antigens, they will kind of lock up and cause blood to agglutinate or clump. Similarly, with blood type B, it has the A antigens, or A antibodies, So if we mix that into the blood type A with the A antigens, the A antibodies will kind of clump up with the A antigens. We'll get agglutination going on. So where are we going with this? Well, when we are given a sample of blood, we need to know and understand how we can figure out what type of blood we happen to be looking at. So in order to do that, we can mix in antibodies to see if antigens are present or not. And to do that, we would use what are known as serums. So there's an A serum, a B serum, and then also the Rh serum, which will determine if the blood type is positive or negative. Serums needs to be tested separately from one another. Otherwise, you don't know which serum is causing the reaction. So in order to get somebody's blood type, you actually need 3 samples of blood from the same person. So what we can do is we can mix in anti-A serum, which has the A antibodies. And if we see clumping going on in this blood sample, we know that this person, this blood, we know that it has the A antigens. So we'll mix in some of the A serum. Guys, mix it up a little bit, and let's see what we have going on after a few seconds. We can kind of see down in the bottom there that it's not nice and watery anymore. The blood has agglutinated, it has clumped up. So right now, before I even tested the B serum or the Rh serum, I know that the A antigen is present in this person's blood. So I now know that this person either has type A blood or type AB blood. Those are the only 2 blood types that have the A antigens. So I'm going to test the B antibody next. We'll see what happens. Again, got to mix it up. Give it a short moment. And no clumping going on. It's still loose and watery. I now know that this is blood type A because it has the A antigen and it does not have the B antigen. Now let's see if it is A positive blood or A negative blood. Again, we're looking to see if those Rh antigens are present. We'll mix in our Rh antiserum. We'll mix it up briefly here, and I can already see that it's starting to clump up. So this is blood type A positive. We'll do a couple more samples. I will mix these up a little bit faster. Okay, So a little mixing. Okay, let's see what we have. Okay, the A serum, still liquidy, so the A antigen is not there. So it can't be type A, and it cannot be type AB blood. Mixed in the B antigen, no clumping going on there. So now it can't be type B blood, which means that we have blood type O here. So is it O positive or O negative blood? Well, that is quite thick. A lot of clumping going on. This is blood type O positive because it has these antibodies. We'll do one more. Okay, so mix in our A serum, our B serum, and our RH serum. Again, mix, little mix. Okay, the A antiserum caused agglutination, so the A antigen is present. The B antiserum caused agglutination, so the B antigen is present. Now I know I have type AB blood. But the RH antiserum, nothing happened, so it is AB negative.